Hello, everyone, and welcome to this webinar. My name is Danny McCarthy. I'm part of the POD Partnership Opportunities and Drug Delivery team. We are the organizers of this webinar. You are watching Overcoming Glioblastoma Drug Delivery Challenges with Nanoparticle Technology. So in this webinar, as nanoparticles can facilitate development and enhance the delivery of a therapeutic, the field of possibilities for difficult to treat diseases is expanding. This webinar will go through a case study and conversation of how Phosphorex applied nanoparticle technology to allow for the injection intravenously for glioblastoma patients. So as I said before, this is part of the, this is being produced by POD, Partnership Opportunities in Drug Delivery. This is a group of pharma, biotech, and drug delivery industry that comes together annually to discuss the latest delivery needs, latest trends, information on deals, and partnering. But this webinar would not be possible without the support from Phosphorex. We're very grateful for them for helping us to put this on. As a schedule of today's events, I'm going to hand it over in a second to Kim Yang from Phosphorex, who will kind of give an overview of Phosphorex and a little bit more about this webinar. Then we'll have a presentation from Dr. Bin Wu and a presentation from Dr. Sean Lawler before we get into a live audience Q&A. This is a live Q&A. Please utilize the chat feature at the bottom of your screen for questions throughout the presentation, throughout the conversation. We will be pulling those, and for any questions we don't get to or any comments we don't get to, we will be passing that along to our panelists. So without further ado, I'm going to have Kim come on screen. Perfect. Hi, Kim. So I'm going to stop sharing and then I'll have you pull up your slides.
Thank you, Danny. Good afternoon. My name is Kim Yang, Marketing Director of Phosphorex. Welcome, and thank you for joining us today to our webinar entitled A Case Study, Nanoparticle Formulation Enables Systematic Delivery of a Therapeutic Agent to Treat Glioblastoma. The purpose of this webinar is to share with you how we use Phosphorex formulation capabilities to enable drug delivery to solve unmet medical needs. This research has been in collaboration with Brigham and Women's Hospital, Harvard Medical School, and Brown University. Our speakers today are Drs. Bin Wu and Sean Lawler. Dr. Bin Wu is founder, president, and chief scientific officer at Phosphorex. Dr. Wu founded Phosphorex in 2005 with the goal of creating a global leader in drug delivery and pharmaceutical formulation. Prior to that, he held senior technical positions in various companies, including UCB, Kimberly-Clark, and DuPont. As an expert in biomaterials, controlled release, targeted drug delivery, and nanomedicine, Dr. Wu published numerous scientific articles in peer-reviewed journals and is the inventor of over 20 granted US patents and more than 30 international PCT applications. Dr. Wu received his PhD in polymer chemistry from the University of Massachusetts Amherst. For Dr. Sean Lawler, who is an associate professor of pathology and laboratory medicine at Brown University, he leads the Brain Cancer Therapy Lab in the Department of Pathology. He's affiliated with the Brown Cancer Center, where he is a co-leader of the CNS Cancer Translational Disease Research Group. Dr. Lawler obtained his PhD from the University of London and his postdoctoral training at UCSF, INSERM, and the University of Dundee. Before faculty appointments at Mass General Hospital, the Ohio State University, the University of Leeds, and the Brigham and Women's Hospital, where he has developed an expertise in translational approaches for brain tumor therapy. Before we start, a brief overview of our company. Phosphorex is a CDMO specializing in particle-based drug delivery solutions. Located in Hopkinton, Mass, and our state-of-the-art facility spans 30,000 square feet with an additional 12,000 square feet of cGMP space coming online in 2024. Our team of technical experts has approximately 2 decades of expertise and experience offering comprehensive service to include lipid nanoparticles for nucleic acid delivery, polymeric nanoparticles for targeted delivery, polymeric microspheres for sustained release delivery. Our capabilities include formulation, feasibility, and optimization, analytical services, process development and scale up, preclinical batch, and in 2024, cGMP manufacturing services. And now I hand the podium over to Dr. Wu. Dr. Wu?
Thank you, Kim, for your kind introduction. Good afternoon, everyone. I appreciate everyone's being here and your interest in our webinar. As we know, glioblastoma is a tough cancer to treat, even though it is considered a rare disease and orphan indication. Over 15,000 GBM patients were diagnosed in the US alone in 2020. There's an increasing trend. We see increasing incidence. It is predicted that by 2030, the 5-year prevalent population in 8 major markets will exceed 300,000 cases. And the 5-year survival is only 5%, making it the lowest survival rate among CNS tumors. As we know, there's no cure for GBM, and treatment options are very limited. Standard of care include chemo, radiation, and antibody therapeutics, but the radiation does not work for larger tumor, and a large patient population have poor responses to chemo, like TMZ. The other challenges include the low permeation of the blood-brain barrier and rapid invasion and progression. Recently, numerous clinical trials have failed at late stages. For example, BMS antibody OPDIVO phase 3 study did not generate the good data as hoped. In summary, GBM remains a cancer with a highly unmet need. Next slide, please. In the meantime, scientists and clinicians have been testing a natural compound called Indirubin and exploring its potential as a treatment for GBM. Indirubin was first identified as an active ingredient in a traditional Chinese medicine against the leukemia. But the history of Indirubin can be dated back to as early as like 6,000 years ago, when people started to use Indirubin-containing plants as a natural textile dye. Obviously, they didn't realize, they were using Indirubin as a compound. In the 90s, an oral formulation of Indirubin was actually approved in China for treating CML. And research has also identified GBM, also identified the Indirubin as an inhibitor for several targets, such as the CDK, GSK3 beta, and STAT. In the 2000s, clinical trials have begun for treating psoriasis using Indirubin. And preclinical data also showing the effectiveness of Indirubin on GBM. And nowadays, people continue to explore the use of Indirubin for new targets and develop new formulations and generating new preclinical data. And next slide. Indirubin and derivatives, as I mentioned, have been found as a valid candidate for effective GBM treatment. Since early 2010, more and more research groups started to realize the importance of Indirubin in treating cancer and other types of diseases. And such as, besides glioblastoma and the pancreatic cancer, breast cancer, and psoriasis, even Alzheimer's diseases. Derivatives of Indirubin have also been synthesized to increase the potency of Indirubin. And it is believed that multiple mechanisms of action may exist, including anti-angiogenesis, anti-invasion, and anti-proliferation. Next slide. Back in 2011, my co-speaker, Professor Lawler, and his colleague, Professor Antonio Hioka, who are the pioneers in the GBM field, discovered that Indirubin inhibited the invasion of tumor cells, blocked the migration of endothelial cells, reduced the tumor angiogenesis, and increased the survival in 3 different GBM animal models. In the meantime, Phosphorex scientists were aware of the importance of Indirubin family in fighting cancer and other diseases, and were exploring innovative ways to formulate Indirubin. So when we developed our nanoparticle formulation in 2016, we reached out to Professor Lawler and Professor Kioka, and we quickly established a collaboration with their group at Brigham and Women's Hospital and Harvard Medical School. Next slide, please. Despite the proven effectiveness against the various cancers and other non-cancer diseases, making indirubin into a drug has been challenging. First of all, indirubin is extremely insoluble. Its solubility in water is in the microgram to nanogram per ml range. So it can only be delivered orally. However, oral formulation has limited bioavailability, as we know, and efficacy, especially for hard to treat cancer like GBM. Injectable formulation obviously is preferred. So researchers have been using DMSO to dissolve indirubin and deliver dosing to the animal. However, it is impractical to use DMSO in drug formulations for humans due to its toxicity. Therefore, it is also a highly unmet need to develop an injectable formulation of indirubin to harvest the positive data on GBM that had been generated. Next slide. For over 15 years, Phosphorex has been leveraging micro and nano encapsulation for drug delivery. Encapsulating therapeutic compounds into biodegradable microspheres and the nanoparticles can lead to many therapeutic benefits. For example, long-acting sustained release, protection of drugs from degradation, and targeted drug delivery. In terms of targeted drug delivery, you can encapsulate a drug molecule into nanoparticles to achieve passive targeting. For example, you can leverage this enhanced permeability and retention effect, or EPR effect, to target tumor, because the tumor tissue is kind of leaky, and the nanoparticles can be trapped in this leaky vasculature, whereas healthy tissue don't have that feature. So therefore, you can have to some degree of targeting. You may also target specific organ, tissue, or cells actively by conjugating a targeting ligand onto the surface of drug-loaded nanoparticles. In addition, nanoparticles can also enhance the intracellular delivery by improving the cell uptake. But today, I really just like to share with you our work on encapsulating encapsulating indirubin derivative into PLGA nanoparticles. And the nanoparticles are referred to as PPRX-1701. And we believe the PPRX-1701 can enable an injectable formulation much needed, and eliminate the need for DMSO. Also improve efficacy with its enhanced solubility and bioavailability. It may also enhance the crossing of blood-brain barrier. Next slide. So to generate the BIA, the indirubin nanoparticles, first we dissolved the tape payload in an organic solvent like a chloroform or ethyl acetate, along with the PLGA polymer. We then mix the organic solution within an aqueous surfactant solution. The surfactant can be polyvinyl alcohol or twin. We then homogenize the mix to form an emulsion, which contains the tiny droplets of the polymer drug matrix. We remove the organic solvent from the droplets to solidify the nanoparticles. And then we remove the solvent. The solvent can be removed by either evaporation or solvent replacement process. And such obtained nanoparticle suspension can be washed with some downstream processes such as centrifugation or diafiltration or tangential flow filtration. And then we can lyophilize the nanoparticles to form the final PPRX1701 nanoparticles. Alternatively, you can use the nanoprecipitation process to generate similar nanoparticles. And such lyophilized PPRX1701 nanoparticles are very stable, and it can be easily reconstituted in a PBS buffer before dosing. I also wanted to add that our process is very scalable and compatible with GMP manufacturing for the clinical manufacturing. Next slide. As you can see, the nanoparticles are quite small. And here, the blank PLGA nanoparticles without the payload had a size of over 100 nanometers. And the BIA-loaded nanoparticles have similar particle size, but a little broader size distribution. Because the entire nanoparticles is only 100 nanometers, and you can imagine the BIA, the indirubin is dispersed, molecules is dispersed in the nanoparticle matrix, and they have even smaller particle size. And that leads to a larger surface area that the indirubin molecules have, and which lead to enhanced solubility and bioavailability. And again, the nanoparticle formulation can be easily reconstituted into an injectable suspension prior to the IV dosing. And so now, so all the nanoparticles were tested in Professor Lawler's lab. Now I'm going to hand the screen over to Professor Lawler. He can explain to you how the nanoparticles were tested and share with you the results.
Thank you, Bin, for introducing all the background to PPRX1701. As the audience will have noticed, that featured some of our previous work using the indirubin drug to treat animal models with glioblastoma that showed some really promising survival data. However, we were, as you mentioned, these drugs are very difficult to work with unless they're specially formulated. And so we were hitting a real brick wall and not able to really take this drug any further on our own without the help of specialized drug development. And when Bin contacted my lab a couple of years ago to explain his discovery and the development of this deliverable formulation of indirubin, we jumped all over it. So I'm going to tell you about some of the studies we've done with that. The paper actually that came out of this will be published later this week on Friday in Cell Reports Medicine. And the story I'm going to tell you very briefly has two elements. One is the testing of PPRX1701 as a deliverable formulation of an indirubin derivative in our lab. And also we explored the effects of the drug on tumor immune interactions. So immune cells interact very closely with tumor cells and are one of the major targets of contemporary cancer therapies, particularly one called immune checkpoint blockade. And so we were interested in exploring the potential impact of indirubin on the host immune system to set the stage for future studies, exploring it as a potential immunotherapy. Next slide, please. So our first experiment was just to ask the question whether PPRX1701, which contains the indirubin derivative called Bayer, behaves the same as Bayer in our in vitro cellular models. So here we show two different kinds of experiments using glioblastoma cell lines. One is a proliferation experiment on the left, and the other one is an invasion experiment on the right, where we put cancer cells or glioblastoma cells into a collagen matrix, and they migrate into the matrix. And what we can see here is a dose response that shows that each of the drugs, the PPRX1701 and Bayer, at the same concentrations, do exactly the same thing. So that's an important thing that we can do in vitro. However, In vivo, it's a different story because we were really unable to deliver the drug without the use of PPRX-1701. So on the next slide, we show our first in vivo experiment. And just to take a moment to explain this to you, the image you see on the right is a mouse, and the target you see is actually a light signal coming from a tumor that we've implanted into the brain. This is kind of thing that work that my lab commonly does. And these tumor cells have been engineered to express a reporter, a genetic reporter that tells us when indirubin and its derivatives are in the cell and inhibiting specific pathways. In this case, it's a GSK3 indicator that drives the expression of firefly luciferase, which then turns on light in the tumor. So our question here was, can intravenously injected PPRX-1701 actually reach the tumor in the brain and cause a biological effect? And as you can see on the right there, after no hours, we can't see the tumor. There's no activation of the pathway. But after 1 hour post PPRX-1701 treatment, we see this strong signal. This indicates that the drug is getting to the tumor. And this is a very important observation in glioblastoma, because one of the major limiting factors for treatment of this disease is the penetration of drugs into the tumor and the tumor microenvironment, because of the strong effects of the blood-brain barrier, which prevent drugs accessing the tumor very well. So if we can go to the next slide, once we'd shown that the PPRX-1701 could deliver Bayer to the tumors in the brain, we constructed a survival study, and we chose to use a model called GL261, and this is a mouse glioblastoma cell line. And the nice thing about this cell line is it allows us to study the interactions of the tumor cells, which are from the mouse, with the mouse immune system, which can recognize it. So this is a very useful model for evaluating immune effects of cancer drugs. And so in this case, we started injections on day 14. So the tumor is well established in the brain at this time. And we injected every 3 days at 20 milligrams per kilogram, which was a convenient dose. And actually, we believe we can get it quite a bit higher. We're pushing the envelope now to explore maximum tolerated dose. However, under these conditions, with this type of dosing, we were able to see a nice extension of survival from 30 days in the control, or a median of about 25 days, in fact, to a median of about 40 days after PPRX treatment, which is a nice robust improvement of survival. And as you can see on the right, we also do MRI imaging of the mice, so we can see the tumors and analyze them volumetrically. And the PPRX treated tumors were clearly growing much slower than the untreated controls. And if we can move to the next slide, this will show some of the immune analysis that we did in these tumors. And so what we found, we used a panel of markers of immune cells, and we pulled those immune cells out of the tumors by dissociating the cells after treatment with the PPRX-1701. And we looked at many markers using a technique called cytoff, or mass cytometry. And this produces complicated maps, as you can see, of heat maps of different immune populations. And what we were able to deduce from this was that there was an increase in CD8 T cells in the tumor microenvironment of treated animals, and a decrease in so-called M2 macrophages. And both of these are strong indicators of a favorable environment for an anti-tumor immune response, which is one of the things that we would really like to achieve in this field, to exploit the immune system as a sort of intrinsic therapy against glioblastoma. So this is very encouraging data to start with. We also looked at a couple of molecular markers, and here on the left we show one called IDO1, which is an enzyme that drives the formation of immunosuppressive metabolites in tumors. And as we can see here, the IDO1 levels are very much downregulated in these treated tumors. If we can look at the next slide now, this shows some in vitro studies just to back this immunotherapy idea up, where we incubated glioblastoma cells, the G9, G68, or G34. These are cells that we've actually isolated from patients who had surgically removed tumors. And these cell lines were tested individually against PPRX-1701 and against activated T cells that we got from healthy donors, which can potentially kill tumor cells. And we also did it in the combination. As you can see, in the red bar particularly, we get a significant decrease of glioblastoma growth when we use the combination of the T cells and indirubin together, or the PPRX-1701. And this supports this idea that PPRX-1701 can lead to a sustained immune response against the tumors. And on the next slide, I show the potential mechanism that we believe is at work here is on the left. And indirubin, we will talk about the mechanism a little bit in the Q&A if necessary, but it's a kinase inhibitor. And we believe it's, in this case, it's inhibiting the JAK kinase, which is downstream of interferon gamma, which is an important pro-inflammatory signaling molecule. And this inhibition of JAK2 and JAK1 leads to a blockade of the upregulation of many downstream genes, and these include IDO1, which is the immunosuppressive molecule that I was talking about. And on the right here, we can see one of our models of how indirubin may be working at least at the level of the tumor immune microenvironment, where interferon gamma, which is produced in inflammatory conditions, can potentially upregulate these proteins like IDO1, which is blocked by indirubin and may sustain an anti-tumor immune response, and also increases the levels of CD8 T cells and decreases the levels of M2 macrophages, both very favorable conditions for immunotherapy. And if we can move to the next slide, considering how we might translate this into patients, we've still got some work to do in the lab preclinically, but we can visualize, potential avenues by which PPRX-1701 could be developed to deliver this drug to patients. On the far right, as tumors recur, this is an opportunity to test many drugs in the dose response, maximum tolerated dose and safety settings when the tumor is very aggressive, very difficult to treat at this point. But also, we could really view this as a companion to standard of care. We have some prior data, which was not shown today, we haven't yet disclosed, indicating that the PPRX-1701 combines well with chemotherapies. And so potentially putting PPRX-1701 upfront alongside chemotherapy and radiation could be favorable. And also, as I alluded to during the immunotherapy discussion, it could combine potentially with immune checkpoint blockade. And these experiments are actually underway in the lab right now to see if we can boost the effects of this type of therapy in our glioblastoma models. And with that, I would like to hand back to Dr. Wu, who will discuss, who will summarize everything for you before the Q&A. Thank you very much.
Thanks, Sean. So as a summary, we successfully encapsulated the BIA synthetic derivative of indirubin into about 100 nanometer sized biodegradable PLGA nanoparticles, PPRX-1701. And these particles can form a stable suspension, which can be IV injected. Therefore, we believe we have overcome the delivery challenge associated with indirubin and indirubin derivatives. And the data showed that the nanoparticles suppress the tumor growth and the migration in vitro. And then these particles are well tolerated, and it could reach intracranial murine GBM. In addition, the nanoparticles significantly shrunk tumor and increased the survival. Therefore, we have demonstrated that the nanoencapsulation can be a viable tool to formulate difficult to deliver therapeutics into injectable formulations for the treatment of hard to treat diseases such as GBM. And the benefit, additional benefit could be improved solubility by bioavailability, enhanced efficacy, and even potential new IPs. And next slide. So I wanted to thank our collaborators at Harvard Medical School and some of the previous employees, scientists who did the work at Phosphorex and Brown University. And again, as Sean mentioned, the work has been officially accepted for publication in Cell Reports Medicine, and you will be able to see them, see the paper this coming Friday. And thank you again for everyone's attention.
Perfect. Thank you so much, Dr. Wu. So I'm going to have Dr. Lawler come back on screen, and then we'll just go through some of these questions. I'm just going to hit stop sharing. Perfect. So we have a ton of questions. So I'm just going to get stuck in, and we'll see how many we can get to it. And again, please ask questions. Please put them in the chat. If there are any that we do not get to, we will be scooping all of these and passing it back to our fantastic panelists. But just kind of going from the very beginning. So a question from Yulia Zabina: would the injection be IV or into spinal cord? If IV, does the compound pass blood-brain barrier as well? Or does it pass well? My apologies.
So actually, I can at least start this one, Ben. So yeah, we've been looking at IV injection, not CSF injection or spinal injection, and we have done some pharmacokinetics and shown that the buyer, the drug that we're carrying in these nanoparticles, does accumulate in the brain and does accumulate in the brain tumor. So yes, these things do cross the blood-brain barrier.
Mm-hmm. Yeah, if I could add also that the nanoparticles, the indirubin itself might be able to passively cross the blood-brain barrier when injected, IV by IV route. But the nanoparticles having, some of the coatings, like for example, like some of the surfactant that coated on the surface of the nanoparticles and could like form like some kind of corona with the ApoE, which could bind some of the receptors on the blood-brain barrier and cause receptor-mediated endocytosis or transcytosis that could enable the passing of the nanoparticles. That's just some hypothesis. We didn't really do, conducted, the serious studies on that.
Perfect. A question from Arthur Klausner. Do mouse glioblastoma models have tight blood-brain barriers? Does the actual human glioblastoma have a tight blood-brain barrier?
Yeah, I can talk to that too. So there is, so the blood-brain barrier, as you know, is very restrictive to the entry of drugs into the brain. The blood-brain barrier in the tumor is different. Yeah, so when you do an MRI scan of a patient glioblastoma, you can see leakiness of things like gadolinium into the tumor. So parts of the tumor are leaky, but the thought at the moment is it's very heterogeneous. So it depends what part of the tumor you're looking at. And we work on a thing now called the blood-tumor barrier, which we're trying to define better as this entity, which is where the tumor meets the vascular endothelium. And we think that still provides a barrier. So if you compare uptake in a systemic tumor versus a brain tumor, even with all the vascular disruption, you still have reduced drug uptake. So we still need ways to get the drug into the tumor much better in glioblastoma.
Another question from Arthur before we pivot to someone else. In terms of GMP production, what scale of LNP formulation or formation was carried out? What is the encapsulation efficiency?
Okay, so first of all, I wanted to clarify these are polymer nanoparticles encapsulating the payload, which is indirubin or indirubin derivative. So it's not a situation of lipid nanoparticles. And the encapsulation efficiency is pretty high, but we don't really need a whole lot of payloads to be loaded in there. Yeah, we can increase the. So there are two things. One is the loading efficiency, but there's also loading percentage. We only loaded about 5%, of the indirubin molecules inside the polymer nanoparticles. Yeah, but if needed, we can increase the loading or we can reduce the load.
For anyone who might have joined late, would you be able to go over the mechanism of action of this again, just in case anybody joined late and didn't see it at the beginning, either of you?
Hang on. Ben, you want to handle that?
The mechanism?
Yeah, the mechanism of action.
Mechanism of action. We mentioned there's a slide. It's actually Sean talked more about this, right? there's a GSK3 inhibition. There's also in other like CDK inhibition, and then we also talked about Sean also talked about the immune the change of a tumor microenvironment.
So this is it's a natural product, and we think it targets multiple pathways that play a role in the hallmarks of cancer, has been just mentioned. So CDKs, which promote proliferation, angiogenic pathways, cytoskeleton. So there's a number of different targets which I think combined lead to these very interesting effects on tumor growth. But mostly protein kinases in our opinion so far.
We have a question from Lynn Yan. Indirubin has poor SciChem properties. Has improvement such as PK been measured, or can improvement of PKs be measured?
We haven't really measured the PK. All right, Sean, do you? I don't remember. We at least we didn't report the PK.
Well, this is part of the work we've been doing more recently. So we've been injecting PPRX-1701 into the tail vein of mice and then monitoring over time, and we do see accumulation in the brain and clearance over a period of hours. We've not compared it with at this point with the nanoparticle, the non-modified version of indirubin. So we don't know if there's a massive improvement in kinetics, but this is the only way we can really deliver the drug using this type of approach. So it's very difficult to compare the two.
We have a question from Darren Wong. Have you looked to see if empty NPs do anything? Also, in your model of how you think these would work, i.e. JAK inhibition, does a JAK-I work in your in vivo model? Have you confirmed that STAT1 phosphorylation is inhibited? I know that there were like 3 questions in there, so I'm happy to repeat.
There were a lot there. So the STAT1 part, in vitro we do show blockade of STAT1 phosphorylation. Yeah, so and prior publications have shown a JAK inhibition by indirubins, including lots of in vitro screens. So it's pretty plausible that is an important target. What were and can you remind me of the other part?
Another question was, have you looked to see if empty NPs nanoparticles can do anything or do anything?
Yeah, so in our initial studies we did that. They have no activity that we've been able to detect. No. And then the other question was about other JAK inhibitors. We haven't compared them side by side at this point, but we do think that it's probably worth doing. But it's a question of time. This is really, we think that this drug has so many different properties that distinguish it from traditional or traditional man-made targeted therapies, that we're really interested in understanding this drug rather than the targeted therapies.
Mm-hmm. Another question. There was one question about sterilization that I wanted to get to. My apologies. From Christian Knopke. I'm sorry if I'm mispronouncing your surname, Christian. How do you sterilize these particles under CGMP?
You can reduce obviously you can, given that the particle sizes are like around 100 nanometers, we can further reduce the particle size, right? So you can effectively filter them, right? Just like what you do with NMPs, and you can filter them to sterilize, the sterile filtration.
Mm-hmm. Anything to add, Dr. Lawler?
I have nothing to add to that.
So a question from Abdul Alam. Does the nanoparticle deliver the entire payload of the drug? And he also asked, what is the zeta potential of your nanoparticles?
So zeta potential part is for PLGA nanoparticles, whether you have a payload in there or not, typically is a negative charged. It is because the PLGA polymer often they have like carboxyl groups on the surface, on the terminal end. So that creates some negative charge. So for example, typically you will see a minus 20 millivolts of zeta potential, and that's what we saw too. And then we also use a pegylated PLGA. Sometimes pegylated could reduce the carboxyl group content, and therefore you have like a lower, less negative zeta potential. We've seen a range of it.
Mm-hmm.
And in terms of delivering the whole payload, that's an interesting question. So we tested that in vitro in one of the first slides I showed, which compared the biodrug without the formulation and the biodrug in the PPRX-1701 formulation. And they looked identical. So at least kinetically in a cell culture system, the PPRX is delivering everything. It's more difficult to test in vivo, but the in vitro data supports that.
Yeah.
There's a question from Paul Sood. Have you determined the fate of the nanoparticles post-payload release?
No, is a simple answer to that question. So we haven't looked at the fate. No, that's a great question, though. And I think would be important to incorporate is in some of the next studies we do. So thank you for that.
There's a question from Shaukat Ali. Have you measured the porosity of PLGA nanoparticles?
We didn't measure the porosity for this specific ones. I mean, we've a lot of experience at Phosphorex in making porous microparticles and nanoparticles, and we have ways to control the porosity. We have a good understanding of what factors would affect the formation of pores. So in this case, we didn't really intentionally to create like porous nanoparticles. And so I believe these particles, although we don't have the data to support, these particles are relatively solid with very little porosity.
And then Lin Yan again asked, can you use this polymer formation to deliver SM to other tissues besides the brain, for example, the liver?
Yeah, I mean, we didn't specifically study the biodistribution of the PPRX-1701. However, based on our experience, the PLGA nanoparticles can a large percentage of them still go to the liver when injected intravenously. And let's say 50–60% go into the liver, but the remaining 40%, 50% will be spread out. Some of them go to the brain tissue, and some of them go into the heart, lung, and spleen. So this could be a way to do extra hepatic delivery, right? If you wanted to deliver specifically, you want to increase the liver delivery, then you can, you can use things like a GalNAc, to attach on the surface, right, of the nanoparticles, and they will drive them more of the particles to the liver tissue and the cells.
A question from Wenlei Jiang. What characterization tests have you conducted for this PLGA nanoparticle? Any stability investigation? Thanks.
Yeah, so we characterized the particle size. Obviously, there are a lot of data we didn't really share here. Just particle size, the loading percentage, loading efficiency, and the stability. We obviously would look at the colloidal stability, right? In the let's say freeze. If you would freeze thaw, that's one stability you would look at. But because the particles are mostly lyophilized, so we would just test the storage, right? In the different humidities, and at different humidities, and at different temperatures. And overall, the particles are very stable because we added like cryoprotectants, and so the colloidal stability is a very good, and we had no problem reconstituting these particles at the time of dosing. Yeah, very long time, we haven't really got to the real like IND stage. And that's not the intention of Phosphorex, right? So we haven't really done like a longer term stability tests.
There's a question from Carl Medvedev. Do either of you have any opinions regarding the planar nature of Indirubin and its contribution to the effectiveness of the treatment?
Could you repeat the question? I didn't get it.
Do either of you have any opinions regarding the planar nature of Indirubin and its contribution to the effectiveness of the treatment?
Yeah, Sean, you want to?
Well, this would be a better question for a medicinal chemist, which I am not, but I do work with medicinal chemists, and we've discussed this, because Indirubins are known, historically as apart from their, potential role in Chinese medicine, traditional Chinese medicines, are also being used as dyes, so this planar chemistry probably contributes to that ability to act as a dye, I guess. But I talked to a medicinal chemist, a very famous one, about this drug, and he persuaded me not to work on it for at least a year or two, because he said, oh no, this the pharmacokinetics will not work. But in fact, what we found repeatedly through doing our experiments with Indirubin and comparing it with other small molecules that target similar pathways, that we always see a much more impressive responses in the biological assays that we perform. I'm not going to give you a chemical explanation, but it's a really great question, that these drugs which really don't possess some of the properties which you would traditionally associate with the man-made drugs, really do seem to do something interesting, and it may be due to these properties. But
interesting. There's a question from Rana Kashfi. What is the drug polymer interaction?
A very good question. So the polymer, I think in order to, in general, if you wanted to encapsulate a drug molecule into like a matrix, like polymer matrix, such as PLGA, then there is one requirement that the polymer and the drug have to really like each other, right? Otherwise, you just get like a phase separation, right? And polymer and you could have like polymer nanoparticles and nanocrystals of the drug molecules separated, right? Coexisted. So in this case, we feel like the Indirubin is reasonably compatible with the PLGA matrix. So that's why they can form the sort of a uniform nanoparticles. You don't see like a separations. There could be some nanocrystals, maybe very small percentage of nanocrystals, separately existing. But we don't know. We didn't really analyze the heterogeneous nature of the formulation. But I think in general they interact with each other well, and forming the stable formulation, uniform, and the molecules is uniformly just distributed throughout the nanoparticle matrix.
There's one question. Given several methods to prepare PLGA nanoparticles, which one is most effective to yield higher drug loading and yield? That comes from Shaukat Ali.
PLGA and what?
Given several methods to prepare PLGA nanoparticles, which is the most effective to yield higher drug loading and yield?
It's a good question, but in order to answer that question, we have to first look at the solubility, the physical chemical nature of the molecule, the Indirubin molecule. So because Indirubin has, not only Indirubin has extremely low solubility in the aqueous media, it also has very low solubility in solvent. It is very difficult to find appropriate solvent to dissolve Indirubin. So we tried, DMSO is the, sort of like a standard solvent for that. But the DMSO is not suitable for like an emulsification process. Therefore, so we use a combination of solvent, like combining DMSO and ethyl acetate, which in which Indirubin has very slight, solubility, very low solubility as well. So we combine the two solvent system, and we were able to create, an emulsion, right? And the other method, nanoprecipitation, and you can get then you just directly use DMSO. You don't need ethyl acetate. You precipitate with the aqueous surfactant solution, and you can get the nanoparticles. In general, the emulsification process can give you is more more scalable. You can make a large quantity. We can make hundreds of grams of them relatively easily. And with precipitation, and nowadays, of course, you have this microfluidics device, which allows you to generate lipid nanoparticles. It can also be used to generate, such Indirubin nanoparticles. But in general, we have a lot of experience in the emulsification process. I think the emulsification process allows for high yield and also high encapsulation efficiency, especially for hydrophobic compound, like Indirubin.
One question regarding, given the wide distribution of delivery of the nanoparticles, was there much tox seen in the PPRX 1701 treated mice? From Sheena Patel.
Toxicity, yeah, we haven't seen any toxicity at the doses. Sorry, I'm stop my video. It's gone shaky, hasn't it? We haven't seen any toxicity at the doses that we use so far. Again, a stronger, an elevated kind of MTD study would be necessary to really show that. But my postdoctoral scientist who did these experiments says that the mice treated with these drugs actually physically look better than the mice that are not treated, surprisingly better. So indications of toxicity, absolutely none. We've done things like sectioning different organs and looking for potential adverse effects. We've not seen anything so far.
Another question: what is the largest scale of PLGA that has been used to date for the preparation of these loaded nanoparticles? From Paul Sood.
So as far as I know, we've developed a PLGA nanoparticles encapsulating various types of therapeutic agents, such as small molecules, peptides, proteins, and even oligonucleotides. So we've made hundreds of grams of scale hundreds of grams of PLGA nanoparticles, and we supported clinical trials, phase 1, phase 2 trials, and so I mean, using some of the continuous process that we developed, you can further increase the scale if you need it.
One question from Christian Nakti again. Does the acidic pH of the tumor microenvironment affect the drug release and particle stability?
Yes, it could affect the release, because the PLGA is a degradable polymer, and the degradation is by hydrolysis. So basically, water molecule can attack PLGA polymer, therefore to degrade it, to hydrolyze the polymer. And whether acid, your strong pH, like a very high, strong basic or strong acidic media would facilitate that hydrolysis or accelerate the hydrolysis. Yeah, so acidic environment, TME environment would probably promote the degradation, promote the release of the payload.
There's a question from Jorge Ruiz. Ben said that nanoparticles could cross the blood-brain barrier due to APOE by transcytosis. Have you considered or measured that binding to APOE by a protein corona assay? I might have butchered the question of that, but have you considered binding to APOA? Yeah,
we haven't particularly done that, but there's numerous publications showing the corona effect, right? When you injected a nanoparticle in vivo in the circulation, there's so many different type of proteins that would like to form layers around the nanoparticles, right? And one of them is ApoE, right? ApoE likes the hydrophobic the entities. That's why the lipid nanoparticle being very hydrophobic, on the surface, and they really attracted a lot of ApoE, and ApoE really, would carry them to liver. That's why you have a lot of liver large presence in the liver tissue. And in this case, the ApoE is just the one factor, because if you use a surfactant like some of the twin surfactant, which has some like a long chain aliphatic component there, and that could attract ApoE, right? And with ApoE also, interact with some of the receptors on the blood-brain barrier and tight junction, right? And causing transcytosis. This is just really we don't really have evidence, so we just say it could be, right? Some such mechanism, in action. Yeah.
So we've just hit the 1 o'clock mark, but before I let you guys go, what is next for this technology? Can you tell us a little bit about what you can expect, or what our attendees can expect from this technology?
Maybe I can say a couple of words, and Sean, if you can add. So Phosphorex is a drug delivery company, but we're also mainly a CDMO, right? We help our clients develop formulations in nanomedicine formulations, right? And scale up manufacturing, to produce preclinical materials and clinical materials when our GMP is officially built in 2024. So we're not really in a position to develop therapeutic products, right? That's not our business goal, right? So we really rely on collaboration and with people like Sean, and to continue the work, right? To continue this research. But we'd be happy to partner with companies, and through like licensing, or through co-development, So we're hoping, and more importantly, we continue to work with Sean, Brown University, to continue our studies. And there's some, as we mentioned, that there's some data showing that we co-development of a co-dosing of PPRX 1701 could actually enhance the accumulation of a chemo drug inside inside the tumor tissue. So that's, we want to continue in that trend, and hopefully generating more data. And on the other hand, we will continue to improve the formulation, the nanoparticle formulation, hoping to further increase the efficacy, and also thinking about in combination therapies, right? Whether in combination with the PD-1 or other checkpoint, and to boost the efficacy.
Yeah, and in the lab, we're going to continue to collaborate with you guys, Ben, and look forward to your new developments. And when we're studying the mechanisms of interaction of these drugs with chemotherapy and immunotherapy, like I suggested, and hopefully, to give us some opportunities to move things forward into patients. And I'd like to thank everybody for their interesting questions, which we will also try and incorporate into our future studies, particularly regarding the fate of the drug in the brain and distribution and those types of questions.
Perfect. Well, I just want to thank both of you so much for taking the time to share your research and to share your work. I want to thank all of our participants. I know that there were questions that we did not get to. We are scooping all of those, and we will be passing those back to our wonderful panelists. But again, thank you so much for taking the time out of your busy schedules, both our presenters and our attendees, to learn about this really incredible work. So again, this webinar's associated conference is the POD Partnership Opportunities in Drug Delivery Conference. Thank you so much for all the wonderful conversations that we've had, all the incredible learnings that you both have shared, and for everyone for participating.